Introduction. Hello and welcome to today's lecture on molecular biology. Today, we will be diving into the fascinating world of protein folding and the role of chaperones in this process. Protein folding is a fundamental aspect of cellular function, and any disruption in this process can have detrimental effects on human health. Now, let's explore the various topics we'll be discussing today in detail. Protein folding is a highly complex and delicate process that necessitates the assistance of molecular chaperones. Chaperones are a class of proteins that assist in the correct folding of other proteins, ensuring their functional stability and preventing aggregation. They play a crucial role in maintaining protein homeostasis, actively supervising proper folding and preventing unwanted intermolecular interactions. When the folding process goes awry, proteins may misfold, leading to the formation of protein aggregates, fibrils, or insoluble clumps. These misfolded proteins have been implicated in numerous human diseases, including neurodegenerative disorders like Alzheimer's, Parkinson's, and Huntington's disease. Understanding the molecular mechanisms behind protein misfolding is essential for developing therapeutic strategies to alleviate these diseases. Molecular chaperones are a diverse group of proteins that can be classified into several families based on their structure and function. Some well-known chaperone families include the heat shock proteins, HSPs, the prefoldin-like family, the HSP70 family, and the HSP90 family. Each chaperone family plays a distinct role in the folding process, ensuring the efficient production of functional proteins. Chaperones are a specific type of molecular chaperone that assists in the folding of newly synthesized polypeptide chains. These large protein complexes provide an isolated environment for substrate proteins to fold correctly. The best-known chaperone complexes are the GRO-L/GRO-S system in bacteria and the CCT/TRIC complex in eukaryotes. These chaperones employ ATP hydrolysis to facilitate protein folding. In addition to chaperones, cells have developed intricate protein quality control systems to ensure the proper folding and degradation of misfolded or damaged proteins. These systems include the ubiquitin proteasome pathway, which targets proteins for degradation, and the autophagy lysosome pathway, which clears aggregated or damaged proteins. Protein quality control systems are vital for preserving cellular homeostasis and preventing the accumulation of toxic protein aggregates. Protein folding follows a specific energy landscape, which includes various folding intermediates and transition states. Studying the kinetics of protein folding provides insights into the factors influencing protein folding pathways, including temperature, pH, and the presence of stabilizing factors such as chaperones or metal ions. Understanding these kinetics helps elucidate the mechanisms underlying protein folding and misfolding. Protein aggregation occurs when partially folded or misfolded proteins aggregate together, leading to the formation of insoluble protein aggregates. The mechanisms underlying protein aggregation involve hydrogen bonding, hydrophobic interactions, and electrostatic forces. These aggregates can have toxic effects on cells and are a hallmark feature of several neurodegenerative diseases. Chaperones not only assist in protein folding but also play a critical role in protein transport within the cell. Chaperone-mediated protein transport ensures the correct localization of proteins to specific cellular compartments or organelles. This process involves chaperone recognition motifs on target proteins, which direct their transportation and proper folding. Post-translational modifications, PTMs, can significantly impact the folding and stability of proteins. PTMs, such as phosphorylation, glycosylation, acetylation, or ubiquitination, can alter the structure and function of proteins. Chaperones often interact with PTM-modified proteins and help maintain their folding and prevent their aggregation. Chemical and pharmacological chaperones are small molecules that can stabilize protein folding and rescue misfolded proteins. These chaperones can be utilized to counteract protein misfolding-related diseases. Chemical chaperones function by directly interacting with the target protein, while pharmacological chaperones enhance the folding capacity of endogenous chaperones. Protein folding diseases are a group of disorders characterized by the abnormal aggregation and accumulation of misfolded proteins. Therapeutic strategies targeting these diseases aim to promote proper protein folding, prevent aggregation, and enhance protein degradation through various approaches. These approaches include small molecule therapies, gene therapies, and modulation of chaperone expression levels. Protein disulfide isomerases, PDIs, are enzymes responsible for catalyzing the formation and rearrangement of disulfide bonds during protein folding. PDIs assist in maintaining the correct redox environment in the endoplasmic reticulum, where disulfide bond formation predominantly occurs. Dysfunction of PDIs can lead to misfolding and aggregation of proteins. Thank you for watching this video on protein folding and chaperones. We have learned that protein folding is a complex process regulated by chaperones and quality control mechanisms. Understanding these processes is essential for understanding disease mechanisms and developing therapies.